And I'd like to begin the JSID guest lecture at IID 2018. The JSID invited Professor Shimon Sakaguchi to give the JSID guest lecture at this memorable IID 2018 meeting. Professor Sakaguchi is currently a professor at the Immunology Frontier Research Center at Osaka University, Japan. He discovered regulatory T cells, which are indispensable constitutes of the immune system for maintenance of immune self-tolerance and homeostasis. He obtained an MD in 1976, a PhD in 1982 from Kyoto University. After postdoctoral training at both Johns Hopkins University and Stanford University, he served as an assistant professor in the Department of Immunology at the Scripps Research Institute. He returned to Japan in 1991 to conduct his immunology research at head of the Department of Immunopathology at the Tokyo Metropolitan Institute of Gastroenterology. From 1998 to 2011, he was a professor at the Institute of Frontier Medical Science, Kyoto University, and served as director of the institute for several years. In 2011, he moved to his laboratory to Osaka University. Professor Shimon Sakaguchi was awarded medal with purple ribbon from the Emperor in 2009, elected as a foreign member of National Academy of Science, United States, USA in 2012, and he received many international prizes, including the Cancer Research Institute William B. Corey Award for Basic Immunology, Tumor Immunology in 2004, the KO Medical Science Prize in 2008, the Canadian Gardner International Award in 2015, and Crawford Prize in 2017. The title of today's lecture is Control of Immune Responses by Regulatory T Cells. Please extend a warm welcome, Professor Shimon Sakaguchi.
Thank you, Professor Morita, for kind introduction. It is my great pleasure and honor to be here and give a talk. So today, the title of my talk is Control No Immune Responses by Regulatory T Cells. And so I believe it is now well established that the normal immune system, it harbors the subpopulation of T cells which are specialized for immune suppression called regulatory T cells. The majority of them are produced by the normal thymus, functionally distinct and mature population. And some of them can also develop in the periphery from naive T cells. They are called peripheral Treg, or in vitro induced, they are called induced Treg. So anomaly of Treg development or function can be a cause of autoimmune diseases and also allergy or inflammatory bowel disease. And also by targeting them, by increasing them, you can establish transplantation tolerance. Or by reducing them, you can enhance immune responses as in tumor immunology. And so the anomaly of Treg, it can be best illustrated by the pediatric disease called IPEX syndrome. IPEX syndrome is characterized by three features. One is autoimmune disease. More than 80% of kids, they develop type 1 diabetes within 3 years after birth. And also almost 100% inflammatory bowel disease and allergy, including dermatitis and food allergy, almost 100%. There's a counterpart in mice called scurvy mice. It has the same mutation on the X chromosome. And it was found around the year 2000 the responsible gene. It was the designated FOXP3. So here we stained for FOXP3 by the antibody we made. So scurvy mice, they don't have FOXP3 positive cells. And if you stain Treg for FOXP3 intracellularly and also CD25 on the cell surface, the majority of FOXP3 positive cells in the periphery, also in the thymus, they express CD25. So if you remove CD25 positive cells from the spleen over the thymus, and then thymocyte, and then transfer the remaining cells into T-cell deficient mice, for example, nude mice. These nude mice spontaneously develop various autoimmune diseases, including here autoimmune thyroiditis, gastritis, and also type 1 diabetes. And the FOXP3, the most important function of FOXP3 is that if you ectopically express FOXP3 in conventional T cells, you can convert naive or conventional T cells into Treg-like suppressive cells. So FOXP3 controls many genes, almost hundreds of genes, but an important function, important role in for suppressive function of Treg, that is the depression of IL-2 and upregulation of IL-2 receptor, CD25. So without IL-2, Treg cannot survive. And also Treg absorb IL-2 from the surroundings. And another important function of FOXP3 transcription complex is that the upregulation of CTLA4, which interact with the CD80, 86 expressed by APC, and downregulate CD80, 86 expression. So for responder T cells, they are deprived of IL-2 and also deprived of co-stimulation. As a result, if the T cell receptor of responder T cells for the MSC peptide, if it is high without co-stimulation, they'll die by apoptosis. If its affinity is intermediate, they'll become anergic. If the affinity is low, they'll stay dormant. So this means that transient suppression by Treg can change the cell fate of responder T cells, either die or inactivated or stay dormant. So today, I'd like to discuss with you the mainly three issues. One is that what determines and maintains the expression of FOXP3 and other Treg signature genes, including CD25 and CTLA4 in developing Treg cells. How can human Treg can be delineated by Treg-specific transcriptional and epigenetic characteristics? And how Treg-specific transcriptional and epigenetic variations contribute to genetic predisposition to autoimmune diseases in humans. So here, if you look at the FOXP3 expressing cells, either natural Treg or FOXP3 transduced conventional T cells, the gene expression pattern is not so similar. Less than 1/3 of the genes are commonly controlled. So this means that FOXP3 is not sufficient for Treg type gene expression. So the lower panel shows the expression of these Treg signature molecules, including FOXP3, CD25, GITR, CTLA4, Helios, EOS. So the FOXP3 transduced conventional T cells, they can express FOXP3, CD25, CTLA4, but not Helios or EOS, indicating that perhaps upstream of FOXP3 gene regulation, the upstream or in parallel, something else, they may control Treg development and gene expression. And also stability. If you compare natural Treg versus TGF-beta in vitro induced, TGF-dependent induced Treg, iTreg, and if you co-transfer with Treg depleted CD4 T cells into lung minus mice, so without Treg, the middle panel, the black line, without Treg, they'll die one by one with inflammatory bowel disease. And if you co-transfer with natural Treg, blue line, you can completely prevent disease development. But the red line, Treg, they are not so good in prevention because lower panel, after transfer, if you trace the expression level of these Treg signature molecules, the Treg, red line, soon they'll lose the expression. So their expression is not so stable compared with natural Treg. So the question is, how do transcription factors and epigenetic changes coordinately control Treg cell development and function? So, of course, epigenetic configuration is perhaps important. And epigenetic configuration, epigenome, this means DNA modification, so methylation or demethylation, and histone modification and chromatin remodeling. So first we looked at any Treg-specific the methylation changes. So this is MEDIF sequencing, methylated DNA immunoprecipitation sequencing. So DNA are chopped down to small fragments and immunoprecipitated by anti-methylcytosine antibody, and we did deep sequencing. So here you see the methylation peaks in Treg and naive T cells. And red circle one, the peak is present in naive conventional T cells, but not in Treg, indicating that this peak is demethylated. This region is demethylated in Treg. And if you look at the whole genome, you'll find 160,000 these peaks, and around them, differentially the methylated, demethylated, mainly demethylated one. That is not so many, around 300 regions, 0.19%. And this peak can be further characterized by bisulfite sequencing. So the peak by MEDIF sequencing, it corresponds to in FOXP3 gene, corresponding to intron 1 region. So each strip, yellow strip means each CPG site. So the Treg, they are demethylated, yellow, but conventional T cells, methylated, blue. So similarly, CTLA4, the region, the intron 1A, and EOS and CTLA4. So you can choose we chose 5 regions among 300 regions as Treg-specific hypomethylation sites. And now we can by using them, we can trace the Treg epigenome changes along in the course of Treg development. So here, FOXP3, GITR, CTLA4, EOS, and CD25. And then the conventional T cells, after stimulation, you don't see any demethylation. But the natural Treg, they stably demethylated. So TGF-beta-induced iTreg, no demethylation. Or FOXP3 transaction, no change. Or memory, or T cell expansion, or Th1, Th2, Th17, no such Treg type epigenetic changes, indicating that a Treg type DNA hypomethylation pattern is very stable and highly specific for natural Treg. And importantly, if you look at the scurvy mice, which don't express FOXP3 protein or messenger RNA. But if you look at the CD25 positive cells, they do show Treg type epigenetic changes, indicating that perhaps epigenetic changes in the FOXP3 expression is kind of independent. To confirm this, we made the FOXP3 transgenic mice and the expressing the GFP. So here, DREG male mice, left side, they are expressing GFP and also FOXP3 protein. But on the scurvy background, meaning that the endogenous FOXP3 is disrupted, only they express GFP without FOXP3 protein. And natural Treg, they show Treg type hypomethylation pattern from the thymus, and it progresses to the periphery. The right side is the kinetics. And FOXP3 null Treg, they also show Treg type the epigenetic changes from the thymus. and the kinetics are almost the same, indicating that really the FOXP3 expression in a Treg type epigenetic changes, they are independent. So they are independent, and perhaps the developing Treg, they recognize the self-ligand, self-peptide MSC in the thymus, and positively and negatively selected. And this TCR signal can induce FOXP3 expression and also the Treg type epigenetic changes. So the question is, what controls the Treg-specific gene transcription and epigenetic changes in developing T cells? So clue is a super enhancer. So several years ago, a group in MIT, Richard Young, they showed that in the genome, there are the genome site where the enhancer is very strong and control the downstream genes very strongly. So these super enhancer sites, you see the dense accumulation of transcription factor binding and histone modification, H3K27 acetylation. This is the permissive the marker for for gene expression, enhancer activation. So these super enhancers may strongly control the Treg and determine Treg cell lineage and also functional stability. So here we looked at the Treg-specific super enhancers. So using H3K, the antibody against H3K27 acetylation, we did immunoprecipitation chip sequencing. And then, so here this is a CD28 and CTLA-4 locus. And conventional T cells, CD28, you see very strong the message. It is called common super enhancer. But the CTLA-4 gene locus, in conventional T cells not activated, but an Treg highly activated. This is called a Treg-specific super enhancer. And left side has small peak. It is small enhancer. It is and specific for Treg. It is called Treg-specific, typical enhancers. So we enumerated the Treg-specific super enhancers, and the lower left, this is the definition of a super enhancer. So the enhancer ranking, and the ordinate is an H3K27 saturation intensity. So it's not linear. At a certain point, the intensity becomes very strong. So the tangent of that curve, if it is over 1, it is defined as super enhancer. So 66 Treg-specific enhancers, and they are importantly, they are associated with Treg signature genes, including FOXP3, CTLA-4, CD25, and so on. And Treg development, in the thymus, so double positive to immature CD4 single positive, and pre-Treg and thymic Treg, T-Treg. So from a T-Treg stage on, you see a FOXP3 expression and also Treg type epigenetic changes. So Treg precursor, in this fax figure, it is defined as CD25 positive, FOXP3 negative, and mature T-Treg as FOXP3 positive and CD25. So right side, you see H3K27 saturation, the chip sequencing. Already from double positive stage at the FOXP3 gene, you see the mark of the H3K27 saturation. It becomes stronger and stronger along the Treg development in the thymus. So similarly, H3K4 monomethylation, it is a marker for poised enhancer. Similarly, you'll find mark at double positive stage, and it will become stronger along Treg differentiation. And also attack sequencing, it's a marker for the open chromatin. So the chromatin start to become open from double positive and opener along the Treg differentiation. And this happens in parallel in not only FOXP3, but also other Treg signature genes, including CTLA-4, the Helios, and then CD25, IL-2 receptor alpha chain. So this means that the TCR signal activate the Treg-specific super-enhancers at FOXP3, CTLA4, Helios, and other Treg signature genes, and the induce transcription and also epigenetic changes. Now the question is, how are Treg specific super-enhancers activated in developing Tregs? And we found that we looked for any molecule which is responsible for these changes. And we did the microarray between Treg precursor versus mature Treg and found that SATB1, this is a genome organizer, is specifically expressed by Treg precursors. So SATB1 highly expressed in the thymocyte and responsible for loop formation of DNA. So right side, the wild type, you see, as I told you, that FOXP3 gene, you see the activation of the H3K27 acetylation. But without SATB1 expression, you don't see such activation. And 66 Treg-specific super-enhancers. No difference in double positive or immature CD4, but the precursor stage of these 66 Treg-specific super-enhancers cannot be activated when SATB1 is not present. And it impairs the Treg development. And so here in the thymus, you don't see CD25 positive, FOXP3 positive cells in the neonatal mice and also 4-week-old mice. And in the thymus, also in the periphery. After birth, for a while, you don't see the Treg cells, and it can cause the very severe autoimmune diseases, just similar to what we see after Treg depletion. So the type 1 diabetes, or the ovary, ovulitis, salivary gland adenitis, and gastritis, and interstitial pneumonitis in the lung. And you can prevent disease development by inoculating normal Treg. And just confirm that how Treg development is affected by SATB1 deficiency. So red square, they are Treg precursors. When you stimulate them in vitro, the wild type, they can give rise to FOXP3 positive cells. And the FOXP3 positive cells, they show Treg type epigenetic changes. But the SATB1 deficient precursors, they are abundant. But if you stimulate them, they don't give rise to mature FOXP3 positive cells, and also no epigenetic changes. So the conclusion is that a certain range of TCR signal intensity or duration can trigger Treg development. Perhaps a pioneer factor, that may be SATB1 plus something, and concurrent activation, it induces concurrent activation of Treg super enhancers at FOXP3 and other Treg signature gene loci, and independently gene transcription and Treg type DNA demethylation, and together forming a stable expression of Treg signature genes, including FOXP3, and functionally stable Treg. Now, this is all based on these results in mice, how we can analyze human Treg. The question is, how can human Treg cells be delineated by Treg-specific transcriptional and epigenetic characteristics? So here, if you stimulate the Treg cells in vitro, human Treg, they can upregulate FOXP3. But unfortunately, in humans, if you stimulate naive conventional T cells, they can also express FOXP3 at a low level, transiently. So meaning that FOXP3 is not so specific for Treg in humans. So how we can define Treg? So this is the CpG hypermethylation, and we compared between fraction 2, the mature Treg versus fraction 6. This is naive conventional T cells, and then red part is Treg-specific hypermethylation. So the left here, in the normal peripheral blood, FOXP3 expression in CD25. It's became linear, beautifully linear. But then they do contain some FOXP3 positive, but non-Treg cells. So we can now, using a CD45RA, we can dissect them into 3 populations, 1, 2, 3. And 1 and 2, they show, fraction 1 and 2, they show Treg-specific demethylation. And also histone, H3K27 acetylation, a kind of marker for active enhancer. So fraction 1 and fraction 2, as red square, are similar and then different from fraction 3 at FOXP3 gene and also CTLA4 gene. So even if they express on FOXP3 and CD25 commonly, but if you look at the epigenome, now you can differentiate real Treg versus non-Treg FOXP3 positive cells. So this is current our understanding that the thymus produces in humans naive resting Treg. And when they are stimulated, they differentiate into fraction 2, the effector Treg. They are expressing FOXP3, CTLA4, CD25, and Treg type epigenome. And they are highly suppressive. And naive T cells, sometimes they can express FOXP3 as fraction 3, but then soon they lose the expression. They are not so stable. And this classification is important for analyzing human diseases. So here, a healthy donor and sarcoidosis patient and active SLE. So sarcoidosis and active SLE in the peripheral blood, similarly, you see the increase of FOXP3 positive cells. But if you look at the Treg subset, sarcoidosis, effector Treg, highly suppressive population, they have expanded, meaning that in sarcoidosis, sometimes you see immune suppression. And this is because the highly suppressive effector Treg, they are circulating almost 10 times larger in number compared with healthy donor. And active SLE, effector Treg reduced, but also fraction 3, which are non-Treg, and producing lots of pro-inflammatory cytokine, they have expanded. And also tumor tissue. And in the tumor tissue, the cardinal characteristics of the human cancer tumor tissue is that effector Treg, they have enormously expanded. So here, ovarian cancer or colon cancer or malignant melanoma, usually in the peripheral blood, effector Treg 2% of CD4, but now in the tumor tissue, 30% or 20% or 40%. And also the colorectal cancer, sometimes the FOXP3 expression, there are some reports that FOXP3 expression is a better prognostic marker for the colorectal cancer. But this is because if you look at the Treg subset, you can resolve the issue. So here, half of the colorectal cancer is a typical Treg type, the effector Treg, fraction 2 expanded. But on the other half, the fraction 3 expanded, non-Treg expanded. So if you compare the FOXP3 low or high, the type A, the fraction 2 expanded, the FOXP3 high, they show poor prognosis. But the type B with fraction 3 expansion, and so non-Treg FOXP3 positive expanded. So FOXP3 high means better prognosis. So this means that really just not look at the full FOXP3 positive cells, but on Treg subset, FOXP3 subset, that is important. So Treg epigenome is the more accurate marker for defining Tregs, especially in humans. So how the human the epigenome, Treg epigenome can be characterized? So again, as in mice, we looked at the differentially methylated regions in human genome. We found that the small percentage of the CPG sites, 0.67%, they are differentially methylated, and especially demethylated. And this region contains FOXP3 and other Treg signature genes. And the FOXP3 gene locus. So here, activated Treg and naive conventional T cells, the methylation rate is very different. So you can extract the variance between these populations. And the high variance region, and then you can do the extract these regions from other Treg signature genes, for example, the IL-2 receptor A, the CD25 or CTLA4. So, but not CD4, no such variance. And with such variances, now you can display the difference by t-SNE plot. This is the technique for reducing high-dimensional data and visualize the high-dimensional data sets. So here are the naive Treg, activated Treg, naive T-comb, and activated T-comb. What's the difference? So here, the red circle of the upper figures. So naive Treg, the red circle, red means demethylated. So after activation, every region becomes demethylated. And lower panel, red circle means in conventional T cells, the Treg regions are methylated. And after stimulation, activated T-comb still methylated. So this is as shown in mice, the Treg type epigenetic DNA hypomethylation. It's independent of T cell activation. So this is perhaps a Treg region. And by further clustering by the technique called the DB scan, now we can have 9 clusters. And then the red circle, that is the Treg region, Treg-specific hypermethylated region, and it includes the FOXP3 signature genes. And if you impose the messenger RNA expression or Treg type, the histone changes, or attack sequencing, open chromatin, the Treg-specific hypomethylated region, they all correlated with these Treg cell characteristics. So by these data all together, we can now calculate the correlation and also the correlation network of these high-dimensional data. And the conclusion is that what is most important is Treg type, naive Treg type CpG hypomethylation, which is the most important one, kind of hub for the major Treg characteristics. As shown here, the naive Treg messenger RNA expression or histone or the open chromatin remodeling. And also activated Treg, similarly, the transcription factor binding, all correlated, all associated with Treg type, naive Treg CpG hypomethylation. So just briefly, last part of my talk is based on these findings, how the autoimmune single nucleotide polymorphism, SNPs, do they locate in Treg-specific super enhancers or Treg-specific demethylated regions? So here, this is super enhancers. We found 19 Treg-specific super enhancers in human genome. And here, the autoimmune diseases, and then the relevant Treg-specific super enhancers. Many autoimmune SNPs locate at Treg-specific super enhancers. The autoimmune disease, including vitiligo, Graves' disease, multiple sclerosis, and type 1 diabetes, and so on. So right-side genes, Treg-specific super enhancers, many autoimmune SNPs, and they locate this in these sites. And the autoimmune, LD means the linkage disequilibrium SNPs. They are also located in the Treg-specific DNA hypomethylated regions. The cluster 3, as shown before, and the cluster 0 is an effector Treg. And the cluster 3 is a naive Treg, and the majority of autoimmune SNPs locate in these sites, these clusters. So this is then now we can fraction 1, 2 the Treg, and then fraction 5, 6 conventional T cells, and each disease, and then each disease has many SNPs. And now you can calculate these SNPs, how much they are demethylated. And then if you look at each fraction 1, 2, and 5, 6, and importantly, the fraction 1, these Treg-specific demethylation has a good correlation with autoimmune diseases. So here, the many autoimmune disease, fraction 1, the highly demethylated the type, and then you see good correlation with these autoimmune diseases, Graves, and celiac disease, and rheumatoid arthritis, type 1 diabetes, vitiligo, psoriasis, and so on. So summary, so development of functionally stable Tregs requires not only FOXP3 expression, but also the establishment of Treg-specific epigenome. And while FOXP3 positive T cells in humans are heterogeneous in function, and functional Tregs can be delineated by a specific epigenome pattern, in particular by a Treg-specific DNA hypomethylation pattern. Naive Treg-specific CpG hypomethylated regions, which are different from T-cell activation-specific ones, control Treg-specific gene expression. And as a result, variations in Treg-specific CpG hypomethylation in naive Tregs contribute to autoimmune disease and susceptibility. And also for treatment, now we are using Treg for the treatment of various inflammatory diseases, including autoimmune disease. So one way is to expand Treg in vivo by inhibiting the effector T cell expansion. And second one is the Treg cell therapy. So importantly, naive Treg, they can be expanded in vitro by antigenic stimulation, and you can transfer back to the patient. And in the future, the ideal way of Treg cell therapy is that if we can convert the effector T cells mediating disease with retaining the antigen specificity, if we can convert them into Treg stably. Stably means not only FOXP3 expression, but also a Treg type epigenome together. Then that is the ideal way of de-establishing self-tolerance. So I'd like to acknowledge my colleagues and recent work on SATB1 that was conducted by Yoko Kitagawa together with Naganari Okura. And recent unpublished the human work was conducted by Yasumizu and Okura and also Morikawa. And thank you for your attention.